Now let's move on to our second group of eukaryotic microbes, the protists. Traditionally, the protist group contains both algae and protozoa. So it's subdivided into two taxonomic categories, or subkingdoms. The subkingdom algae, which we'll only cover briefly because they generally don't cause human infections, although there are a few exceptions we'll take a quick look at. And then the subkingdom protozoa, or the animal-like protists. Although these general types of microbes are now known to occupy several different kingdoms, we're going to retain the concept that protists are unicellular, or sometimes colonial, eukaryotic organisms that lack true tissues. Let's look quickly at the algae. Usually they're recognized by their macroscopic members, like the seaweeds and the kelps, and they can vary in length from a few micrometers that we see in the Spirogyra here, which is a filamentous form that has spiral chloroplasts in it, up to 100 meters in length, such as the giant kelps. Algae are photosynthetic protists, generally. We'll see one exception coming up here shortly. And they're classified by their pigments. So we'll see red algae and brown algae and green algae. The green ones have a predominance of chlorophyll, but the others have other pigments that lend to their naming. There are some forms of photosynthetic protists that cause red tide that we see in shellfish that prohibit us from eating it, and some other blooms we'll take a quick look at. In the figure in the middle here, you can see diatoms. Diatoms are tiny, beautiful algae that are characterized by silica cell walls. We think of them as little glass houses, very ornate glass houses with a variety of different forms. This figure on the right is of particular interest. It's an image of Pfiesteria piscicida, one of the dinoflagellates that causes another sort of bloom. Red tide is also caused by dinoflagellates, but a different form. We'll look at that shortly. These are free-living, but known to be parasitic to fish. They release a really potent toxin that can kill the fish and sicken humans. It causes skin lesions and neurologic symptoms in the fish, as well as humans that are exposed to contaminated water and eat the fish. It's a newly identified algal parasite and has been reported over the past several years along the southern Atlantic coast. There is a particularly large outbreak as a result of a huge bloom of this particular dinoflagellate. These blooms have been traced back to the nutrient-rich agricultural runoff. The extra nutrients cause a bloom in the algae. When those algae are consumed by fish, they release this potent toxin, and then that potent toxin is passed up through the food chain to us. You can find a plethora of information on this topic online if you'd like to go a little further and explore this perhaps for your final project. Here are examples of some of the classes of algae that we see. There's Phaeophyta, which are brown. These are macroscopic algae, or kelp. Rhodophyta, red algae. These are also macroscopic. This is the class of algae that provide agar, the nutrient source that we found in many of our petri dishes. Chlorophyta are the green algae. They're both macroscopic and microscopic. Then Euglenophyta, they're motile, usually single-cellular, green, animal-like algae. So they have characteristics of both animal cells and algal cells, so they belong in a unique group themselves. Now, the dinoflagellates are of particular interest. We just took a look at Pfiesteria piscicida, but red tide is also caused by a species of dinoflagellates called Carina brevis. These cause the Florida red tides. More recently, we've discovered a new group called the Prototheca. These are green algae, but they contain no chlorophyll and they don't perform photosynthesis. They're saprobes, meaning they either obtain their nutrients from their host or by decaying organic matter. Ciguatera is another serious intoxication caused by these algal toxins that have accumulated in fish, such as bass and mackerel. Cooking doesn't destroy the toxin, and nor is there any antidote. So if you eat contaminated fish, the symptoms could prevail for days or even months. They involve things like vomiting and neurological symptoms like tingling fingertips. So we've covered 3 conditions that are caused by algae, but in general, algae don't cause too many human medical conditions. As we begin to look at the biology of protozoa, you'll recognize that they are the most vivid group of microorganisms, especially as characterized by the way they move around. Their name comes from the Greek roots meaning first animals, but they're far from being simple or primitive organisms. In fact, they're really complex and quite fascinating. Remember that we've grouped these protozoans just for convenience rather than any sort of relatedness. They're grouped based on similar physical characteristics, not genetic relatedness. There are about 65,000 species that are mostly single cells. Sometimes they form colonies, and they have startling properties when it comes to movement. In fact, often we group them by their mechanisms of locomotion. Most protozoans are harmless, free-living inhabitants of water and soil. Few of them are parasites. However, these few are responsible for hundreds of millions of cases of human infection each year. So again, most are single cells. Sometimes they form colonies. They contain all the major eukaryotic organelles except for chloroplasts. The cytoplasm is usually divided. There's an ectoplasm layer and an endoplasm layer. The ectoplasm is the clear outer layer, and it's primarily involved in locomotion, feeding, and protection. So it may form flagella or cilia, or even pseudopods to allow amoeboid motion. Sometimes these cilia aid in feeding. The endoplasm layer is a granular inner region. This is where most of the workings of the cell are housed. The nucleus, the mitochondria, sometimes food, and then contractile vacuoles are located in this endoplasm portion. Protozoans have no cell wall as we typically think of a cell wall. Some are cased in hard shells, but this is different than a cell wall. So in general, they have some flexibility in the movement of their cell membrane. Their cell membrane is typically the outer boundary, and it regulates the movement of food and wastes and other cellular secretions from the cell. The cell shape can remain very constant, or it can change constantly, as we'd see in amoebas. And the size ranges from 3 to 300 micrometers, but on occasion we'll see very large protozoans, like giant amoebas and some huge ciliates that could be up to 3 to 4 millimeters. That means you can actually see them with the naked eye swimming around in pond water. Let's take a quick look at a video of some common protozoans.
Protists are abundant in water and on land. Most, like this amoeba, are single-celled. Its false feet or pseudopodia are used for feeding and locomotion. Volvox is a colony of single-celled organisms. Special reproductive cells give rise to new individuals. Paramecium is covered in tiny hairs called cilia that beat in time to produce movement. It uses a structure called a vacuole to engulf and digest food. Paranema, however, can manufacture its own food and uses its whip-like flagellum to propel it forward.
So as vast as this protozoan group is, we're only going to be looking at some selected medically important protozoa. Keep in mind there are many, many more. Basically, we have 4 groups, and we group them based on their method of motility, their mode of reproduction, and then stages in the reproductive life cycle. Let's begin by looking at the methods of locomotion or motility. Some unusual species will move by gliding and twisting. We have 3 basic means: the pseudopods, flagella, and cilia. Pseudopods are what we see in amoeboid motion. You just saw some in the previous video clip. These amoeboid structures or pseudopods can also serve as feeding structures. Another means are flagella. A protozoan may have only 1 or several flagella. Some are attached along the length of the cell and move by the undulation of a membrane. In addition, there are cilia. Mostly these are distributed over the entire surface of the cell, and they form characteristic movement patterns. Let's take a look at movement with flagella and cilia, as well as some of the other functions that they have.
These Chlamydomonas swim very fast using their 2 flagella. At high magnification, the 2 flagella become visible when a cell is embedded in mucilage, slowing their activity right down. Like many algae, Chlamydomonas has an eye spot which responds to light and affects the way the flagella beat, steering the cell toward or away from it. Many protists have flagella of different length and function. This euglenoid, called Phacus, is swimming in the direction of its longer flagellum. These relatives of Phacus also have two flagella which function differently. One beats in a coil around the cell. The other, as in Phacus, extends forwards. Other algae have more than two flagella. This cell has 8, inserted into a deep pit. It normally swims very rapidly and had to be slowed down to have its picture taken. Large protists like Paramecium swim rapidly using hundreds or even thousands of flagella, or cilia as they are usually called. They are very agile as they search amongst the pond debris for food. Beating of the cilia is coordinated, and the direction of beating is reversed when the cell changes the direction it wants to go. This cell, like the last ones, had to be slowed down in a sticky solution so that it could be photographed. Many protists also use their cilia to create currents for feeding, carrying nearby organisms close enough to be eaten. These algae are too big for this protist, but show the currents clearly. This large protistan cell, called stentor, feeds like a vacuum cleaner. A circular array of cilia around the flattened end of the cell sucks nearby organisms into a conical mouth where it can check out what it has captured. It is feeding here on green algae. Notice that it is quite selective about what it eats, swallowing some of the captured cells and rejecting others.
Now, if pictures are worth 1,000 words, videos must be worth 1,000,000. This one I thought I'd include just because it's an amazing video of a paramecium magnified 400 times. It's just beautiful. The life cycle and reproductive mechanisms of protozoans vary from very simple to complex. The trophozoite stage is the active life stage, where the organism is metabolizing, growing, and reproducing. But it requires plenty of food and moisture in order for the protozoan to remain active. Some protozoans are capable of entering a dormant stage when the environment becomes less favorable. This stage is called the cyst stage. So some exist only in the trophozoite stage, and others alternate between trophozoite and cyst. In the cyst stage, the trophozoite cell curls up into a ball and the ectoplasm secretes a tough, thick cuticle. This allows the encysted protozoan to survive adversity, but it still is able to be dispersed in currents in the air. This could be an important factor in the spread of diseases like amoebic dysentery. When favorable conditions return, the cyst breaks open and releases an active trophozoite. Because life cycles vary from simple to complex, this affects the mode of transmission to the hosts. For example, trichomoniasis is caused by the protozoan Trichomonas vaginalis. It does not form cysts, and thus it requires intimate contact and plenty of fluid for transmission to a new host. However, Giardia, the microbe responsible for many of our illnesses, drinking water from high mountain streams that's been contaminated by cows, is caused by the protozoan Giardia lamblia. These do form cysts, and they're very hardy cysts, and thus Giardia is readily transmitted from contaminated water and foods, even if the foods have become dried, because this protozoan becomes encysted in order to survive the adverse conditions. Now let's take a quick look at the 4 different groups of protozoans that interest us in this course. First of all, we have the Mastigophora. They move using flagella. As we've already established, protozoans are categorized based on how they move and what their reproductive cycle looks like. So we'll begin with Mastigophora. They use moving flagella, a whip-like motion. They have very complex life cycles and many parasitic species that we're interested in. For example, Tympanosoma and Leishmania are important blood pathogens that are spread by insect vectors. We'll take a look at those in more detail later on. Giardia, again an intestinal parasite spread in water contaminated with feces. And then the Trichomonas are parasites of the reproductive tract of humans, and they're spread by sexual contact. The next group we consider are the Sarcodina. These ones move using pseudopodia. They consume food by means of phagocytosis. There are only a couple of species of particular interest to us. Entomoeba histolytica are pathogens of particular interest to humans. The third group we'll consider are Ciliophora. These ones use moving cilia. Most of them are free-living, and the paramecium that we saw earlier is a great example of this. Ciliophorans have a macronucleus and a micronucleus. They reproduce by conjugation. One of particular interest to us is Ballantinium coli. The apicomplexans, also known as sporozoa, are non-motile protozoans that are obligate parasites. Ones of interest to us are Plasmodium vivax and Toxoplasma gondii, which causes acute infections or toxoplasmosis in humans, and it's acquired from cats and other animals. So again, as we try to identify protozoans and cultivate them, we'll consider the shape and size of the cell, the type, number, and distribution of the locomotor structures, the presence of special organelles and cysts, how many nuclei they have. Much of this can be done by simply observing them under the microscope. Most protozoans can be cultivated in artificial media or in laboratory animals. In this table, we can see some of the important protozoan pathogens that we'll be covering throughout the remainder of the semester. Take a quick look, but fear not, as we go through the different systems and collaborate on our wikis, we'll cover many of these pathogens.